Hello and welcome to Nikolay's genetics lessons, and today is going to be my second video about gene nomenclature. And before I will give you a new information today, I want to review previous information because I'm going to make about 15 or maybe 20 videos about gene nomenclature because this topic is very interesting. And at the beginning it might think that for example, naming of proteins, genes, messenger RNA, and copy DNA is confusing, but later you will find that just looking at the name of the gene, you would be able to tell a lot. Because there are about 50 different rules how we name genes, and knowing those rules would help you to navigate through different names. And once again, genes, messenger RNA, and copy DNA. If it is human gene, we use capital letters. All letters have to be capital, and we italize. If it is going to be animal gene, then only first letter would be capitalized, and all the rest would be in lowercase and would be italicized. And proteins would have the same names as a gene that encodes this protein, and would be capitalized in both humans and animals, and wouldn't be italicized. And today I want to give you one more rule. For example, if you would see such a name, you still wouldn't be able to tell whether this name stands for the gene name or messenger RNA or cDNA. So in order to specify this information, we just include this information in front of the name. So for example, that this is messenger RNA. And if we want to specify on which chromosome this gene resides, we include also number of chromosome using 3 letters acronym for chromosome, so C, H, R. We do not put period here or any dots, and also include chromosome number. For example, Down syndrome cell adhesive molecule, this gene can be found on the chromosome 21. Now if you would see such a line, you would be able to decipher it as we have Down syndrome cell adhesive molecule messenger RNA, and the gene that produces messenger RNA resides on the chromosome 21. And of course, a messenger RNA would be without introns, so this is going to be messenger RNA that only includes exons. And because all letters in the name of the gene are capitalized, you would know that this is human gene and not animal gene, where we only capitalize only first letter. Now I want to explain a couple more rules about gene nomenclature. For example, right now these names wouldn't tell you much, but believe me, in just a couple minutes such names would tell you a lot. For example, if you would take a look at this picture, what you would see animal cell, and here is the nucleus, and we also would find mitochondria and cytosol of the cell, and each mitochondrion would have a circular chromosome that can be present in many copies. So this is not different chromosomes. This is just a copy of the same chromosome that can be present in from 2 to 10 copies per one mitochondrion. And on each such chromosome we can find, for example, in humans 37 genes. Actually, in order to operate, mitochondria would need much more genes, much more proteins. We can find within each mitochondrion about 1500 proteins, but those proteins would be encoded by nucleus. and only 37 genes left on the chromosome that we can find in the mitochondria. And whenever we want to name such gene, this would start with MT. MT stands for the mitochondria. And then hyphen, because in many genes we would see hyphen, but if hyphen would go after these two letters MT, that means that this is mitochondria. Next letter also important. Next letter after hyphen can be of two kinds, whether it can be T or any letter. And T stands for the transfer RNA. And next letter after T stands for the amino acid that this transfer RNA would be charged with. For example, if we would take a look at this table of the amino acids, we would find what this C stands for, because names of the amino acid has full name, 3 letters name, and 1 letter name. And if we would take a look, we would find that C here stands for cysteine. So now we know that this is mitochondrial gene, tRNA, that would be charged with cysteine. So this is going to be the first type of genes that we can find in mitochondria, and second type of the gene, as I said, may start with any letter, and that means that this gene would make some kind of protein or enzyme. Now let me remove this table and bring to your attention this table that I took from Wikipedia, and here you would see many mitochondrial genes. Out of 37, 22 would start with T after hyphen. That means that this is transfer RNA. So as you see, transfer RNA everywhere here, and next letter also would stand for the amino acid. And also in this table you would see L and H for each gene. What does it mean? That means on which strand of the DNA we would find this gene. It can be heavy chain or light chain. Mitochondrial chromosome is circular, and it is double-stranded, of course, and if we make a cut somewhere here, for example, we are going to get linear chromosome, and we can also separate two strands of the DNA, and because one strand would have more cytosine and thymine, for example, once again, cytosine would base pair with guanine and thymine with adenine, and these bases would have 2 rings and 1 ring. For example, cytosine would have 1 ring base, thymine also would have 1 ring base, but guanine would have 2 rings and adenine also would have 2 rings. So these bases would be heavier than these bases. And if by chance we would have on one strand of the DNA more cytosine and thymine, and on the other strand of the DNA we would have by chance more guanines and adenines, then this strand would be heavier than this strand. So scientists, when they separate two strands of the mitochondrial DNA, they get one strand that is a little bit heavier than the other strand. So we call one strand heavy strand and another strand we call light strand. If we'll count all the genes that is present on the heavy strand and light strand, we would find that there are more genes that is present on the heavy strand of the DNA. And about 1/3 of mitochondrial genes we would find on the light strand of the this DNA. 22 of these genes would code for mitochondrial transfer RNA, and the rest would code for proteins and enzymes. Now I hope you would be better able to understand such abbreviations and also would be able to use them, and at least would know where to look up for what amino acid transfer RNA, mitochondrial transfer RNA stands for. And this is all for today. Thank you for your attention. Please subscribe for my new videos that I post almost every day. Thumbs up if you like this video. Please write your comments, questions if you have any. Share this video with your classmates, and see you in the next video. Goodbye.